Hi again, my name is Rodrigo Nofre. I'm a postdoc fellow here at K-State Plant Pathology Department. And today I'm here to talk a little bit more about ascomycetes. And as you can see, I have some wheat plants here in my hand. These plants were saved and cultivated by Dr. Eric Dwolf. He's a professor here in the Plant Pathology Department as well. So many thanks to Eric. So for today's class, my goal is to give you a little bit of taste of that lab activity that we would be doing in a normal year, right? But because of COVID, we're trying to adjust and trying to expose you as much as we can with all the tools that we have in hand. So for today's class, we're going to be looking into wheat powder mildew. This disease occurs in all over United States and also Canada. Powder mildew, it's one of the biggest problems in several crops, especially in the specialty crops such as rose, strawberries, vegetables. Wheat powder mildew, it's caused by a fungus called Plumeria gramineus form speciale tritici. So now we're going to talk a little bit about the symptoms, right? So powder mildew on wheat and other cereals as well, and grass, are very remarkably similar. In this case here, powder mildew in wheat may infect all aerial portions of the plant. So all above ground can be infected, and that infection can happen after emergence, right? As soon as the seeds start germinating, if temperatures are favorable for disease development and the pathogen is present, right? So it can cause infection and the symptoms can be visible. The symptoms will be most prevalent on the upper surface of the lower leaves, right? And the reasons because they're like on that lower part of the plant where there's like more humidity and the temperatures are very favorable right there. So that humidity and temperature is just the perfect storm and perfect conditions for this disease. The pathogen grows, as I mentioned before, entirely in the superficial part of the leaf, right? And the only thing that actually gets into the epiderm cells, it's called austoria. And it will be through the austoria that the pathogen will absorb the nutrients that it needs from the plant. This disease can occur in patches in the field, but as it sporulates, it can spread across many areas, right? So as I mentioned, the colonies will initially look white and very fluffy, as you can see on this top part here. But later on in the season, or as the pathogen will mature, it will turn dew or gray, dark brown in color, right? So after the pathogen infectant sporulates, those places where infection happened can turn yellowish, or we can see some chlorotic patches that will normally appear on that leaf surface directly opposite to the mildew colonies. And as I mentioned as well, this disease in our part 1 of ascomycetes, right? So if you remember very well, powder mildews, they produce Cleistothecia, right? And in case of wheat powder mildew, it can also produce Cleistothecia as well, which will contain the asc, and then inside the asc, the ascospores. I'm going to try to demonstrate here one activity that we would be probably doing in the lab if we were having this in person. And I hope, let me know if you have any questions, and I hope you guys can visualize what we're doing here well, and yeah, and hang in there with me. So what I'm going to start doing is selecting a colony that looks young and that mycelium and sporulation is very fluffy. And I found one right here. I'm going to cut that out. Going to put it here on the table. Just going to cut it out here. Try to show it to the camera. You can see there's a little colony right there in the center, right? So what we're gonna do now, we're gonna use a glass slide. We're gonna put it here on the table. We're gonna need a drop of water not too much water, right in the center, right there. Then now we're gonna get that leaf that we have right here in the center. We're gonna use a transparent tape, and I'm gonna tape that leaf and gently pushing on the top of the surface. Right now I'm gonna remove the tape, and the idea here is just to get all that mycelia that was present in the leaf into the tape. Okay, not sure if you can see, but it's right there. So now what we're gonna do is we're gonna bring that tape to the our glass slide, and we're gonna tape it over that drop of water. And this is how it will look like. Sometimes when we're doing this in the lab, and several times, it is good to highlight or put a little circle surrounding that spot that you really want to see under the microscope. So here I'm just like putting a few spots that I would like to show you right there, and now we're going to look into the microscope. Okay, so after taping the leaf, right, we place a drop of water and put that tape with the mycelia and all those spores into our glass slide with that drop of water. And our idea was to try to get all the conidiophore and some of the conidia that was being produced in that chain, right? So as I mentioned, powdery mildew here will have Conidiogenes thallica. So all those spores are gonna be being formed from the part of the conidiophore. So we're gonna start looking here with our lowest magnification, as we should always start in the microscope. And now we're gonna switch, which was 440, we're gonna switch to 100, our next lens. And now, as you can see, the first one, we couldn't see much, but we were able to locate where our sample was, or our point of interest, right? So here we start seeing some of those spores. We can notice their shape a little bit more clear now. We can see some chains, which was our main objective here, was to see some of those beautiful chains, as we can see here right on the center top of this video. However, I'm not still not quite happy. I think we can amplify a little bit more. So now we're gonna switch here to a higher magnification, and now we're seeing those chains even better. As you can see here, those spores are being formed, and as they get to the edge, they're being in a mature stage higher than the ones that are in the base. So the ones that are in the base, they're younger and they're developing, and as they get old, they're gonna be on the edge, and that's when they're gonna be broken out and released with rain splash or wind or any mechanical movement on those leaves. So I'm trying to give you an experience here of how it would be in the lab. What we're seeing here, it's wheat powder mildew, conidiophores, and the conidia they're all being produced from that conidiophore by Conidiogenes thallic, which is the spores are being formed through that conidiophore. We can see in the conidia the point of attachment. Between them there's a little brighter point, and several of them. I'm just gonna try to move around a little bit more so you can have an idea of how many we were able to obtain from that tape. So this is a very easy and quick way to see and visualize powder mildew structures under the microscope. Okay, so we're here back with the wheat plant with powdery mildew, as you can see, and we're going to be going over just a little bit more about what are the favorable conditions for powdery mildew in wheat development. So this disease, it's strongly tied to the weather. If we think about mild winters, this will favor early and severe mildew development in winter wheat areas, right? So watching the temperature, it's always very important when we're thinking about disease, especially the powdery mildew ones. The spores, as I mentioned before, are spread by wind, and they don't spread too far. They're relatively to spread within short distance. If we have cool and humid, dry conditions will favorable infection, and this pathogen in specific does not occur in free water, right? It doesn't require free water for the infection. Rain, in case of we have a rain event, that water will wash off the spores, but disease can rebound quickly depending on the intensity of that rain. If we have prolonged periods of cool and humid weather in the spring, this also can allow disease to reach the flag leaf and perhaps cause some yield loss. There's a lot of research within wheat powdery mildew, and most of it refers to when temperatures are above 79 to 81. This temperature range will slow down the disease progress and disease development. Okay, so we're here in our last part of our lab, and we're going to be going over a few management strategies that our growers are available for our growers. So number one is resistance. There are several cultivars that offers complete or partial resistance to this disease. And if you're choosing a cultivar with complete resistance to powdery mildew, it might not require a fungicide application. So two fungicides application is also recommended for powdery mildew control. There are several fungicides available in the market. Triazole seems to be the best ones for suppressing this disease, and you need to be in the watch, especially if you're planting a susceptible variety and the conditions are favorable for that disease development. So in case you see that early on, it might be an option for you to suppress this disease. And three, it would be some cultural methods to suppress, right? As I mentioned before, high dose of nitrogen can increase the disease severity and incidence of powdery mildew in the field. So make sure that you're not using high dose of nitrogen, and also avoid high plant populations. So when you have high plant populations, what we're going to have is you're going to be favorable in that disease development and also infection, because high plant populations, we're increasing the humidity among plants, and also we're going to be creating a microclimate that will be favorable for disease development. So we have fungicides that are available for our growers, resistance within the cultivars that can be used, being complete or partial, and also some cultural controls, which is the nitrogen and high plant populations. With that, I say thank you very much. I hope you enjoy our virtual lab, and I'm looking forward to hear more from you, and let me know if you have any questions. You can always send me an email. Thank you very much.